Dr. Richard Frye, thank you so much for inviting me to speak today. I really appreciate it, and I hope that the talk is informative. Let me pull up my slides. Okay, today I'm going to talk about the personalized multidisciplinary approach to evaluating and treating autism spectrum disorder. I'm Dr. Richard Frye. I'm the Chief Scientific Officer of the Autism Discovery and Treatment Foundation, and I practice at the Roswell Medical Center in Phoenix, Arizona. Today I'll talk about some treatments for autism. These are not approved by the FDA in the United States. They're used off-label, so there's nothing wrong with that, but it's important that anybody that uses them should know the risks and benefits for each type of treatment. So autism has increased dramatically over the last 20 years. It seems to go up, at least in the United States, but there's similar trends around the world. Since 2000, it's increased almost 400%. What is autism spectrum disorder? Autism spectrum disorder is when we have symptoms. It's diagnosed by particular symptoms. We used to think of it in the DSM-4 as having deficits in social abilities, communication abilities, and repetitive behaviors, and these different categories of PDD and OS, Asperger's, autism. But now we actually look at it as just a spectrum, and as far as autism, we do not differentiate now between social and communication issues because they're so intertwined. And what is the crux of autism? I mean, people a lot of times think it's a language disorder where, children can't talk, but the fact is it's a communication disorder. That is the ability to communicate with another person. I think it's best emphasized when we look at something that's very important in infancy called pointing. Pointing is the way that an infant it's the first way an infant will communicate with their parents to show them something or indicate something they want. And there's two types of pointing. Of course, there's proto-imperative, which is when you point to something you want, and this is simple pointing. And sometimes children with autism will develop that. And then there's what we call proto-declarative pointing, and this is really more social pointing, where you point to something because you want to share that experience. You want to show somebody that something is important to you and they should look at it. So as I mentioned previously, we had different categories of autism, and then now we have the DSM-5, which has collapsed social interactions and social communication together into one category, since it's so hard to really disambiguate them, and have added to the restrictive and repetitive behaviors sensory issues too, which are so common in autism. And so now we have these two domains that you need to have symptoms, both in the social communication domain and in the behavioral domain of restricted interest and repetitive behaviors. And one thing that I really made clear that a lot of people don't quite understand is that you not only have to have the symptoms, but they have to function, they have to interfere with your life in some way. So you could have all the symptoms of autism, but if you're a normal functioning person who is able to do everything they need to do, you really don't meet the criteria for autism. And so when we look at autism, We have our core symptoms, which you just talked about, the social communication impairment and restricted and repetitive behaviors, which we see over here on the left. However, we have other comorbid conditions that come along with it. We have specifiers for the diagnosis of whether it's intellectual impairment, language impairment, medical conditions, genetic conditions, environmental factors, and such. And then we have very common associated symptoms: irritability, impulsivity, anxiety, attention deficit, hyperactivity, executive function, and learning disorders. So what I'm going to propose is, well, when we look at the severity of autism, we rate autism in 3 levels: requiring support, requiring substantial support, or requiring very substantial support. So, as I mentioned, you know, if you don't, these symptoms don't interfere with your life, really you don't meet the criteria for autism. So I think one of the things you can think about is, what is the goal? Not so much to completely eliminate symptoms or characteristics of autism, right? Because some people say, oh, I have autism because I have this characteristic or that characteristic. But what we want to do is get the person to be so high functioning that they require no support. And they are severity level 0, and essentially the autism diagnosis disappears. So what is autism? How does it affect the body and the brain? What causes autism? So what we found is a very multi-dimensional disorder with many different factors. We have factors that affect specific organs, excuse me, such as the brain, the immune system, and the GI system. These seem to be all involved for some reason. And then we have many environmental factors that make things better or worse. These you could think of, there are a wide variety of environmental factors: social, psychosocial factors, educational factors, sensory factors, dietary factors. Many times children need special diets because they're very sensitive to allergens and such. Allergies, chemical sensitivities, and environmental toxins. And on the other side, you know, autism doesn't just affect these organs. It affects every cell in our body. So we know that there's systemic disorders that affects the mitochondria, redox metabolism and the ability to make glutathione, methylation metabolism, which controls turning genes on and off, and then genetic disorders where there's changes in the genes, which are in every cell of your body. As far as the brain, we know that there's multiple disorders. There's an imbalance in the excitatory inhibitory cortical balance, such that there's more excitation in the brain. There is seizures and epilepsy we see. We see abnormalities in the basal ganglia circuits, which control movement. We have abnormalities in what we call the amygdala, which controls emotions and the autonomic nervous system in the fight or flight. You see the fight or flight system many times really turned up to 11 on kids. And this is through the amygdala. This is through anxiety and control of the autonomic nervous system. And then we have neurotransmitter imbalances, many of them, including norepinephrine, serotonin, dopamine, and acetylcholine. So one of the key pathologies in autism is this hyperexcitability of the brain. And the brain is the cortex, the outer part of the brain where you do your thinking, is very well organized usually. And it's organized into these columns, which you see here. And each of these columns has a very specific organization. In the middle, you have a pyramidal cell, which is excitatory. And then surrounding it, you have inhibitory cells, which calm down that excitatory cell once it fires. Because once you get excited, you have to calm down. When we look at the pathology of autism, of the cortex, if we looked at the cortex under the microscope, and these are two pictures, we see something that's very striking. If we look down at the bottom of the typically developing individual, if we look at their cortex, we can see these mini columns. We see that they're actually very well organized. These little black dots, the cell bodies, so different neurons that are here, which are very well organized in these columns. Now we'll go over to the individual with autism, and we see these columns are not all that well organized. They're squished together mostly, and the reason why is because those inhibitory cells that surround that excitatory cell are not there. So there's no break to that excitation. There seems to be many GI disorders. This is a study that was done by the Autism Treatment Network, which is a large network in the United States of autism centers. And we can see that when we look for different GI disorders, constipation, 35%, diarrhea, 5%, gastroesophageal reflux, 30%, anyone with any type of gastrointestinal problem, 58% they found in their clinic. So that is big. What's really interesting is that many of these GI disorders go unnoticed, and there's certain behaviors that we have to look for that will tell us whether there's issues going on. Sometimes, for example, for reflux, we'll see tilting of the head to the side and arching the back, which sometimes people take as just some type of stereotype movement. Sometimes a child can't explain abdominal pain, so they push on the abdomen when it's when it's uncomfortable, or they refuse to take foods in. Interestingly, many behaviors that are associated with autism are thought to just be irritability, have been associated with these GI problems. For example, screaming, sobbing for no reason, whining, facial grimacing, unusual posturing, arching of the back, self-interest behaviors. There's many cases where self-interest behaviors were found to actually be solved by solving the GI issues. Sometimes this takes a gastroenterologist to actually look very carefully, but sometimes this can just be constipation, which can really make the child feel terrible, and we have a lot of irritability and sometimes self-injury. Then there's the human microbiome, and those are the trillions of bacteria that live in our gut. We really did not appreciate that until recently, that these bacteria that are there are probably doing something, and they are actually part of the physiology of our body. And many times these bacteria can be imbalanced, and that can cause issues in the body. We've linked autoimmunity and activation of the immune system to autism. Interestingly, this started a while ago, back at the turn of the century, where they were taking medical history, and they noticed that many children with autism, that there was a history of autoimmune disease in the family, particularly on the maternal side. Really, the idea that there was some immune activation actually in the body for children with autism was really proven and really solidified by this paper by Dr. Zimmerman, Pardo, and Vargas. And what they did is they looked at autopsies. So they took individuals with autism that had died for other reasons, and they compared them to other individuals that had died for other reasons besides brain injury or such, and matched them very And they took a look at their brains. And what they found was very striking. So if you look at panel A here, this is a micrograph of the cerebellum. And what you can see is that it's very nicely organized. You have this white matter here and the outside, and there's this very distinct border between the white and the gray matter. And in the gray matter, you can see all these tiny little dots, which are individual neurons that are organized next to each other and very well placed. And then we go to B. We look at the cerebellum of an individual with autism. And what we see is that, well, this white matter here isn't so nice. There's pieces, little divots and such. There's holes in it. You look at the gray matter, and then there's actually, it seems to be holes in the gray matter all over the place. And there's no distinct border between the white and gray. It's not very distinct. So they asked, well, why is this? And one of the things they did was use something called a stain to find out if there's any inflammatory components. And all these little brown dots are markers of inflammation throughout the cerebellum. Other studies have looked at autoantibodies, that is, antibodies that attack our cells, and to see if there's any autoantibodies in autism. And a wide variety of autoantibodies have been found to attack many elements of the nervous system. One of the very important components of autism, where we see abnormalities, is in the metabolic systems, what we call one-carbon metabolism. One-carbon metabolism is essentially denotes folate metabolism and the folate cycle. And the folate cycle is attached to something called the methylation cycle, which is extremely important because the methylation cycle creates this thing called SAM, which is our methyl donor that is used when we have to do a certain chemical process called methylation. And methylation is very important for turning genes on and off, right? You may wonder, well, you have the same genes in your liver cells as your kidney cells. Why is one a kidney cell and one is a liver cell? And that's because certain genes, they all have the same genes, but certain genes are turned off that are not needed. And if you can't do that, the cells are not going to work correctly. So methylation is connected to something called glutathione metabolism. And glutathione is the major antioxidant of your body. What is needed is these precursors from the methylation cycle to make glutathione. And we find that actually the body of kids with autism under normal conditions cannot make as much glutathione as they need. And so this production of glutathione actually disrupts methylation. Methylation is tied to the folate cycle, which is very important. And it's very important to realize that there's different types of folate. We think of folic acid as our folate, but folic acid is the synthetic form of folate and cannot go directly into this folate cycle. It has to go through something called dihydrofolate reductase. So that's the enzyme that activates it. One of the issues is that has an upper limit of how much it can work. And it can only activate about 400 micrograms of folic acid a day. So you cannot treat a folate deficiency with folic acid. You just can't get enough converted into the body to be used. That's why you have to use other folates. The one that we use many of the time is glucovorant calcium. These abnormalities were really discovered by Dr. Jill James. She has an interesting story. She used to be a cancer biologist and then serendipitously got involved in autism. And she showed that all of these different metabolites that I showed you on the last page, and these different cycles, were significantly different in kids with autism versus controls. These biomarkers have not just been shown by one lab, but several labs, many labs across the world over and over again. So we know these abnormalities are real. We know that's in many tissues of the body, including the brain. We've looked at postmortem brain to show that they're there. And we know what happens is if you have a decreased glutathione, which is your major antioxidant, there's actually abnormalities. So you find oxidative changes to DNA, protein, and lipids in kids with autism. Of course, there's the genetic abnormalities that we talked about a lot. There's estimated that about 5% have cytogenetic problems, there's chromosomal problems, there's Fragile X. Depends on where you are in the world. It seems to vary a bit, but it's probably about 1% of autism. Rett syndrome, maybe up to 5% of girls. Then we have something called copy number variations, where little pieces are missing or duplicated. And then we have single gene mutations. And I have to update the slide because we just presented a paper where we show that if you look very carefully, up to 50% of kiddos with autism will have a primary diagnostic variant that is related to autism, but they're almost all what we call de novo. They're not inherited. They're actually new mutations that occurs. So genetics is definitely something that's very important, but it doesn't count for all of autism. And it's very complicated. And so this is a diagram of those cycles we talked about just before, the folate cycle, the methylation cycle, and glutathione production. And there's many different variants, that is, we call these polymorphisms, common variants. And we know that they're overrepresented in autism, but the polymorphisms within themselves can cause autism because they're very common changes. But what was shown in this paper by Dr. Jill James was that if you look at the combination of these variants together, you can see that the risk of autism goes up dramatically. So if you have a polymorphism in the cobalamin transporter and the enzyme that is important for processing and eliminating neurotransmitters, you can see that the combination of those two can increase your risk of having autism 7-fold. So it's the combination, most likely, of these different polymorphisms. And this shows what we really think is happening is that there's a polygenetic landscape, that it's not just one gene. It's combinations of genes with environment. And this is a really nice example of how environment can interact with genetics. In this study, Art Bordeaux, who is a famous geneticist from Baylor in Texas, from Baylor University, looked, first he looked to see if he can find in a cohort of individuals with autism whether he can find a gene that accounted for their autism. And overall he couldn't. When he looked at it, just simply he couldn't. But then he looked at specific subsets and he found out that indeed there was the gene that makes something called carnitine, which is a very important vitamin, was abnormal, but only in families where there are male multiplex families, that is, that families that had multiple males with autism. And so what does this gene do? Well, it is important for making carnitine, but carnitine is a conditional essential vitamin. That is, you get it from your diet. mostly. But if you don't get it from your diet, you actually need to make it, and you have this enzyme to make it. So, in order for this to cause autism, you not only have to have the mutation, but you also have to have a diet that's deficient in carnitine. So this is just a diagram of how we go about treating kids with autism, and we, at baseline, collect history, behavioral questionnaires to actually tell us the different characteristics, their different symptoms. We review these symptoms and order a test that we need. And then we select the treatment. So, how do you select treatments? Well, it's best to do treatments at least, one or a few at a time. So you have to consider what treatment am I going to select first. So we usually select one that addresses the most problematic symptom, the symptom that's most likely to improve other symptoms. So that's where you have to think about how things are related. So if GI distress is causing irritability, you want to treat that first. The things that are most likely to respond to treatment, of course, you want to treat the things that are easy to treat first, because that's where you can make the most headway. And we always were evidence-based, so we always pick the treatment that has the most evidence. And how do we know about what's going on in the body? Well, there's many biomarkers that allow us to tell what is going wrong in the body. And they're divided into many different categories. We have prenatal history, genetic biomarkers, neurologic biomarkers, but most of them have not been validated. There's only a few that have been validated, and one is the transmethylation, transsulfuration, that is the methylation and glutathione biomarkers have been validated over and over again. Mitochondrial biomarkers have been studied. There's something called the maternal autism-related autoantibodies, and these are antibodies that are in mom. It's estimated that 20% of the mothers with children with autism have these antibodies that will attack the brain, the fetal brain, during development. The folate autoantibody, which is very important for getting folate across the nervous system. And then, as far as autonomic biomarkers, heart rate variability is what we see as a validated biomarker. Again, we can talk about the importance of this cycle, the methylation cycle, the glutathione metabolism, folate cycle. And so why this has been validated, so after Dr. James found that this was abnormal, she teamed up with Jürgen Hans, who was able to take those numbers and show that you can use these numbers to actually separate those that have autism and those that don't. So diagnostically, you can use this with a 97% accuracy. And what's most important is you can fix these things. So in one of the first studies done, methylcobalamin or methyl B12 was injected 3 times a week along with folic acid, and we looked at the change in the Vineland adaptive behavior scale before and after, and here we can see the change in months. So in 3 months of treatment, you can see that language improved anywhere from 6 to 8 months, and socialization improved up to 12 months. And this is on average. This is the maximal extent. One of the things that's really important about fixing this reaction, which I didn't mention before, is that when you make glutathione down here, it's made of cysteine, which is very important. It comes from this methylation cycle, but it's also made of glutamate. And glutamate is the major excitatory neurotransmitter in So you actually reduce the excitability of the brain by making glutathione. The mitochondria is extremely important. The mitochondria is the powerhouse in the cell. We have anywhere from hundreds to tens of thousands of mitochondria in each cell of our body. Mitochondrial diseases were first described in 1988, which really in medical science is not that far away. Because they make energy, we see dysfunction in what we call energy-dependent tissues that use a lot of energy, including the neurologic system of the brain, the gastrointestinal system, and the immune system. And those are the exact systems that are dysfunctional in kids with autism. In 2012, we published this paper where we looked at the prevalence of mitochondrial problems in autism. We found something very interesting. We found that the incidence of prevalence of mitochondrial disease, which is a very specific form of mitochondrial dysfunction, was 5%. And this is thought to be very rare. So this is a very big number, percentage of anything to be 5%. But then we looked at these biomarkers that the mitochondria wasn't working, and we found that their elevations, up to 30% of kids had these abnormalities. So it seemed like the mitochondria was not working in many of the kids, but it didn't constitute mitochondrial disease. So we hypothesized there's other types of mitochondrial dysfunction. And in this study, they looked at, and this is from the Mind Institute, they looked at lymphocytes from children with autism, 10 children, 10 controls, and they actually found that 80% of the immune cells, the lymphocytes in an individual with autism, showed abnormalities in mitochondrial function. We looked at those that were diagnosed with mitochondrial disease in autism to see if they had certain characteristics, and in fact they did. They were more likely to have neurodevelopmental regression, seizures, motor delays, so gross motor delays, which is very somewhat unusual in autism, GI abnormalities, and interestingly, only 23% of them had any type of genetic disorder that could explain their mitochondrial disease. There's many different abnormalities we found in the mitochondria, the different complexes, complex 1, 2, 3, 4, 5, have all been shown to be decreased and sometimes increased in function abnormally. We've seen abnormalities in the fatty acid oxidation pathway. So we have our biomarkers. We know there's some genetic abnormalities associated with them, and we have treatments. And as I mentioned, that we hypothesized that there was some unique type of mitochondrial dysfunction in kiddos with autism, and this is just briefly what we found over 15 years of research, that kids with neurodevelopmental regression, their mitochondria, instead of working very poorly, is actually working overtime. And these are diagrams from the seahorse, which we use to look at the respiration rate of the mitochondria and immune cells. And we see that down here in this orange line. So is what the typically developing sibling had, and up here is what the sibling with neurodevelopmental regression and autism had. And you can see that the respiratory rates, how hard the mitochondria was working, is about twice that of their sibling. Also, one of the things we have linked this mitochondrial function to is prenatal exposures. We've found that actually air pollution and nutritional deficiencies can change and be related to changes in mitochondrial function long term. And one of the things that we can look at is to see all the factors that have been linked, prenatal factors that have been linked to autism, all affect the mitochondria. The question is, if you have mitochondrial dysfunction, what do you do about it? The purists, mitochondrial geneticists many times say when you treat the mitochondria, if there's mitochondrial disease, But in this paper, we argue that that's not true, that even if the mitochondria is not working for some other reason, you need to treat it to improve function. The studies there haven't been that many studies, but this is one of the studies that actually systematically used mitochondrial cocktail, certain supplements that support the mitochondria, and found kids that had mitochondrial dysfunction and autism. And they showed by using the simple cocktail, in 3 months they were able to improve mitochondrial function and to also improve behavior. This is one of the other really important findings, these maternal antibodies. So these are antibodies that are in mom, which will cross the placenta, and they attack these specific proteins in the fetal brain. The big one that we find is abnormalities associated with folate autoantibodies. There was a disorder called cerebral folate deficiency described in 2005. And many of these kids were described to have features of autism. And what happens in this disorder is that folate doesn't get into the brain. In order for folate to get into the brain, it has to be attached to a transporter, the folate receptor alpha. And the folate receptor alpha essentially pulls folate into the brain. But many children, up to 75%, have this antibody that binds to this transporter and prevents it from working. So when we found out or realized that this may be related to autism, we did our first study, and we showed that 75% of kids had the antibody. Either 60 had the blocking, there's two antibodies, they had blocking antibody, and 50% had the binding antibody. And so this is treatable. This is treatable with leucovorin, which is a special type of folate that can get around that block. And this is the first double-blind placebo-controlled study. There have now been 4 additional studies confirming this work, controlled blinded studies. But we found that leucovorin, or at that time we called it high-dose folinic acid, significantly improved verbal communication, and that the folate autoantibody predicted those that would respond. What's very interesting and compelling is that these same transport mechanisms are on the placenta, and that we find this autoantibody in mothers of children with autism. So this may be affecting things before birth also. And Dr. Rainmakers, who originally described the cerebral folate deficiency from his clinical experience, he came out with this diagram to show that depending on which of the parents has the antibody, it can be related to the prognosis of the child. This is one paper, if you're interested in these biochemical pathways, that we published in 2014. That is a nice review of the different metabolic pathways and the different treatments. Also, we know autism is related to many different deficiencies, including carnitine and zinc and other B vitamins. And here is just a diagram of the other biomarker for autonomic activation that we see as heart rate variability seems to be significant. And the higher the heart rate variability, the more healthy you are. And in this study, this is a study where we used a transcutaneous neurostimulation, and we measured anxiety. And you can see here the red line, what happened when the patient had to sit in the clinical room and actually do a battery of tests. That heart rate variability went down when the stimulator wasn't turned on, so that's your sham. And as soon as we turn the stimulator on, take 2, you can see what happened. It improved heart rate variability and anxiety. So I'm just going to skip that. This is kind of what we have in our clinic. And I think this is really important to follow kids along with specific symptomatic questionnaires. And we use the ABC and the SRS, and we look at these each time they come into clinic, so we can actually follow their symptoms to see if their symptoms are getting better or worse. And this is always important because you want to know, which way you're going with your treatments. So with that, I'll thank you for your attention, and I hope it was informative.